Okay, Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. This is really a very interesting topic for biology students and for general interest for all, I think. Biotechnology, as the name indicates, I think, can you guess what does it mean, biotechnology? Would you like to just tell me what does it mean? This is a combination of two words, right? Bio and technology. So as the name indicates, we are using biological system, okay? And there are some overlapping terms: biogenetic engineering, biomolecular technology, biotechnology, genetic engineering. All these are the same terms or techniques that we're using these days. And they are very common, making drugs, for example, enzymes that we need, and the industries, even the vaccine that we have these days, the corona vaccine, is also a product of biogenetic engineering or biotechnology. So you are using biological system, you can manipulate, because we know the genetic makeup of the genetic information, the entire genome, the entire genetic information that was revealed, I think, in 1970s. It started from there. The boom started, and people are taking much interest. And of course, there's also commercial pressure. We need things. You could see the fruits production, the drugs that we need, the vaccine, and the farming is also revolutionized due to biotechnology. So in this chapter, I think they have given a very good figure here: enzymes to treat disease. As you know, there are many diseases where we definitely need enzymes to treat. One of the most common hormone or enzymes that we are dealing with is the lactose intolerance. You know, this lactase, people cannot secrete lactase, so they cannot tolerate, they cannot digest easily dairy products, milk or yogurt, you know. So that is also one of the things that we are facing. But they have given in the trigger in the opening the Gaucher disease. They are saying that this disease, as you know, is deficiency of the enzyme which is dealing with the fat molecules, the lipids. So we have a recessive allele. Recessive allele means the allele, the gene, which is responsible for the production of this enzyme, which is taking care of the fat molecules, are not synthesized due to some reason. You need normal allele. The allele is the pair of gene. So as you know that for each enzymes are basically proteins, right? So for the proteins, we have genetic code that is responsible for controlling the expression or synthesis of that particular proteins. If the genes are recessive, I mean, they are not normal, so you won't have that enzyme functional. And that's why people have problem with the fat metabolism. And this is called Cashier disease, okay? And they are saying that usually these cerebrocytes builds up in the body organs, which is very dangerous if they build up in liver and spleen. And there's one of the things that you might see in obesity. If you are eating lots of junk food and not restricting or adopting normal lifestyle, and you know, the fat accumulation will take place and things will build up in liver, and there is a damaging also. So remember, this enzyme deficiency, which is causing Gaucher disease, will build up more fats in the liver and spleen, and which is very dangerous. So finally, they found, the researcher, just that there's an enzyme which is missing in this disease. So, and they tried to do some research studies. If we can use that enzyme, would it be beneficial for those people with Gaucher disease? So would it replace this deficiency? Would it work in those patients or not? So as you know, that this is the initiation of bioengineering or biotechnology era. This is the beginning of the bioengineering. So they isolated an enzyme from the human placenta that was extracted or isolated, and the results were promising. It showed very good data, very good effect, and very good control. So as you know, if you are using this for many patients, of course, you cannot use normally extracting this enzyme from the placenta. So you need a commercial production for this enzyme so that you can use it for many patients, right? So probably it might work in 50 or 100 patients for research purpose. But if you want to treat these patients of Gaucher disease with this enzyme, you should have large amount of enzyme commercially available. Okay, so that is how in 1980s scientists found a way to make this enzyme by genetic engineering. So if you know exactly what gene code is responsible for the production of this enzyme, if you can take this code from normal people, and then you could use in biotechnology, we are basically using microorganisms like bacteria and fungus to produce drugs and enzyme, because these are small organisms, they don't need much space, and their requirements are very less, right? So it doesn't have any ethical problem as well. At the same time, you can, in the multiplication process is very fast. That is why you can produce this enzyme through through genetic engineering. What do you mean by genetic engineering here? That you will take the code of that gene and you can incorporate that code into bacteria. Okay, so this bacteria will multiply, and bacteria is also synthesizing proteins. As you know, that DNA is common, right? The DNA that we have, our cellular DNA, and also the bacterial DNA are the same building blocks. The codes are the same, right? So you can easily incorporate that particular code of the gene, that gene which is responsible for the production of enzyme in the bacteria, and that bacteria would multiply, so that can produce large amount of enzyme that can be isolated. Because when bacteria is growing in a medium, you provide, big fermenters are used or big vessels are used so that bacteria can replicate, and then you can isolate the enzyme. Okay, this is how they are doing it commercially. So till here, I think if you have any question, you can need, but we will go through some practical examples here. So they have mentioned here the application of what exactly is biotechnology, as I mentioned, is the use of living organism or biological system. Is it clear? Oh, yeah, it's clear. Yeah, I'm kidding. Can you just justify it? Yeah, okay. Right. So what exactly is biotechnology? As I mentioned, is a combination of bio and technology. This technology is used using biological system. Mostly we are using bacteria and yeast. Yeast is a fungus, or bacteria we are using for production in drug industry, and also in the agriculture industries, in farms, and other commercial industries. We are using these biotechnological techniques. Usually the term is used when microorganisms are involved, when plants or animals are used to produce something that we need, right? So the beauty of this technology is that you can selectively produce things that you want. For example, in case of diabetes, insulin. Insulin is produced by pancreas. So people who are diabetic, the type 1 diabetes, there is the lack of insulin release because pancreas is damaged. Pancreas is no more working. So these people can survive only if you will provide them insulin. How you can kill so many animals and then you extract the insulin from pancreas, this is very difficult, and it would be very, very expensive as well. You won't be able to manage patients with this traditional way. So what you will do, basically, you will take the gene of the insulin, which is the gene which is synthesizing insulin. So you could, by genetic engineering or genetic engineering, you can incorporate, or you can make it enter the bacterium, that particular gene, and the bacteria would synthesize insulin. So this insulin can be easily, as you know, that E. coli is the commonly used. This is the name of the bacteria we are using for production of insulin. Okay, so that is why we are using microorganisms, because they are very efficient and they can produce things in a very fast way. So there are certain reasons, and I think there are also questions that we'll go through, inshallah, in the end of chapter questions. But why we will be using microorganisms for the biotechnology or genetic engineering? So as you know, that they are very small. This is one of the reasons. They have mentioned some of the justification that why we would use bacteria and fungi, because they are small organisms and they are easy to grow in the laboratory. It does not take up much space and they reproduce very quickly. These are the characteristics, right? Of course, you need to take a lot of care and protection measures to avoid infections and spread and resistance also in other microorganisms that will be problem for humans. Uh, so this is another way they are saying that there's no ethical issues. Ethical issues are still there because you cannot do it in open spaces. And of course, you should have proper labs, you should have proper protection, and you should have proper scientists who are expert. These are the ethical issues. But it does not have those ethical issues that we have with animals or humans. Of course, you cannot use each drug, all drugs on a human is an experimental animal. You cannot use it. And even if you're using small laboratory animals like rats or mice or rabbits or guinea pigs, you still have standardized way. There's called ethical issues. You cannot kill animals just like that, and you should have minimum use of animals, and you must take care that the environment is safe by using these biological samples, from the animals. So these are some ethical issues. And of course, animals are difficult to grow, like monkeys, dogs, rabbits, guinea pigs. It needs big houses and lots of human manpower and resources. So it's really expensive. Genetic engineering, the beauty of genetic engineering, you don't need much spaces. You still can control the production in a limited space in a very efficient way. So this is the beauty of genetic engineering or biotechnology. Okay. Yeah, they have again mentioned here that as we know that bacteria are totally different from human, but the DNA structures, genetic code is almost the same. That is why we can use bacteria to incorporate our particular DNA, a particular gene, and the bacterial DNA that will synthesize that particular enzyme or proteins that we want. So, and then they have further given, I think, explained some of the microorganisms, especially the yeast, which is a fungus, right? These are single cell, and they have also mentioned the structure. I think you all already know about the structure: cell wall, cell membrane, the nuclear material is here, the vacuoles here, cytoplasm, and all the stuff, right? And you can see the growing procedure here. They are saying just if you give them proper environment, they will grow. So the budding, you can see the small budding coming up, they are separating. So they are replicating very quickly by way of buds. This is the normal way of replication for fungus. So what does it do? Yeast is a common, what is yeast? That fungus which is very common, present in soil. It's present also on leaves and vegetables and fruits. It's also on the skin, right? And this is why when you are uh, grinding the wheat and then you're preparing the dough by mixing water. So there's also the yeast which is working. So what does it do? Yeast is basically doing a fermentation reaction. This is called fermentation, and we call it anaerobically. Make it sure that you keep it. This anaerobically does not need oxygen. You know that? So that is, it does not need any oxygen. Okay. So what does it do? Yeast, that fungus can split glucose and cause breakdown of the glucose into ethanol and carbon dioxide. So this is something that we are using in baking industry and also making biofuels. Biofuels, as the name indicate, we have fossil fuels, right? By the fossil that we are using a refinery. You have in Saudi Arabia big refineries in Jubail and also in Dammam and also in Yambu, and they are using these fossil fuels a refinery to refine and make the petrol or gasoline for the engines, right? And they are saying that to cut down on the fossil fuels, there's an alternative by making use of biotechnology. They are using maize, which is corn, corn, maize, right? This crop is common also in the Western countries, and we do grow it in different in the Indo-Pak and other regions. So maize is one of the crop that is used in the process of they are so one of the things you need to know that what exactly is the main constituent, main content of the maize seeds. What does it contain? Anyone would like to share? What does it contain? Maize. We are using amylase enzyme. Okay, so I disclose it. I mean, this is another hint. Amylase is working. Yeah, starch. Exactly. Very good. Starch. Yes, amylase is working on starch, right? So maize is containing mostly starch. So what will happen if we mix this with amylase, or amylase is activated? So what will happen? So it will convert the starch into glucose, right? It's releasing the glucose. So that is actually amylase is breaking down the starch to the glucose, and then yeast, which is present. If you, you know, with the, as I mentioned, this is very common in our environment. This is fungus. So it will start the fermentation process. So by this way, glucose will be broken down to ethanol, which is extracted from the mixture by distillation. I'm sure you know the process of distillation.
Yes.
What is that process? Would you like to please say something about it so that people can know?
It's basically a process in which you heat a specific mixture of liquid to different temperatures, so you can get different um, amounts of solution at different temperature.
Yeah, what is the process of distillation? How does it takes place? How does it take place, the process of distillation? You are right. Yeah, the mixture of solution, different solution, you want to purify them, you want to isolate them, right? Yeah, making use of the boiling point, because if the solution is there, each solvent has got different boiling point. So it's evaporating in sequential way. And that is why you can evaporate that, and then you can do the condensation process. So which one is evaporating first, you can select the temperature. You could use also other parameters to control it. In here, you can easily, even if there's other mixture, you can isolate or you can extract ethanol very easily. And it is, you know, that alcohol burn very easily. That can also be used as a energy for the engine, but is a fuel, right? But it does not have that much energy that we have from the fossil fuels, the actual gasoline. So sometimes they are saying you might mix that with the biofuels can be mixed with the gasoline, right, to have more production, and that might help in the industry. Although there are lots of limitation, of course, it's not going to work. That's why we are not using this as a main alternative. We are dependent on fossil fuels. And now things are diverting towards the bioenergetic car, electricity cars, and all those run by the chargeable batteries and all the stuff. So yes, but we are still dependent on fossil fuels, because the alcohol which is produced even through maize is not a good source of energy for engines. And they have further given some discussion here that even if you're growing crop of maize, this is an alternative to biofuels. Remember, bio mean biological, your natural produce. So that is the natural produce that we are growing. So you get the fuel from there. So the main source here, is they have mentioned here, is maize, right? So maize could be used as a source for biofuels. But they are saying that if it can also help in removing the carbon dioxide. You are burning fuel. What is produced? Carbon dioxide, right? So that carbon dioxide is taken up by the plant. So this is in one way better, because carbon dioxide is used by, of course, for photosynthesis. They need to produce glucose, right? And that's one of the reasons glucose is then stored in the form of starch. So this is a mutual benefit, you could say, to the environment, and the plants are using carbon dioxide, and then we are using these plants for energy production. At the same time, you cannot use maize as an alternative source, because then you have to grow crops on a very large area. So it's possible that it will compromise, you know, you will compromise other produce. You won't have space for other vegetation and other fruits and other production. So you need to pay for that. So the price will be, of course, very high if you're using this. This is, I mean, general discussion. I'm not sure how important is this for your exam, but you should keep in mind what exactly is biofuels, right? So you will understand the way of biofuels. So you start with the maize, which is containing starch. Starch is broken down by amylase. Amylase is converting starch into glucose, and glucose is then fermented by the action of yeast, which is fungi, carbon dioxide, and ethanol. And ethanol is then by distillation, removed by distillation. And this ethanol, and they have further discussion, though we don't have much commercial use of it, but yes, we do use it for in case of medicine, preparing medicine and other industries to use this by natural alcohol. Making bread, I think in baking industry, yeah, we use the fermentation process very often, right? As you know that the bread, which is mainly made of cereal crops or wheat flour, this is the main, right? The grains and the wheat seed that we are grinding, and we made the wheat flour. And you remember, in wheat flour, what does it have? What does it contain? Starch, right? You agree with me? She is saying that starch. Starch is the main constituents, right? So flour is containing starch, lots of starch, and also protein, which is called gluten. Remember, when you put water, you mix water with the flour. What happened? You could see a sticky material. So it's absorbing water. This protein is easily sticking together. If you stretch the dough, which is made of mixing flour with water, so you cannot break it easily, right? It stretches a little. So it's because of the gluten proteins. So they can swell. And to make the bread, to make the bread, the flour is mixed with water and yeast to make. So yeast is already there. If you are making bread, of course, mixing water with your hands, you are introducing yeast as well, which is the fungus, right? So what will happen? The moment you add water to the seed, the amylase is active, right? So there's a how even the seed germination, if you remember, we discussed in seed germination. So the initial storage, because you may not have much water, much energy for the seedling, for the plant, the small plant which is growing, they need the energy which is stored in the seed, and that is in the form of starch. And the starch is then by amylase action is broken down into maltose and glucose. So the yeast can further work in an anaerobic way, which is called anaerobic respiration. So it produces bubbles of carbon dioxide. Okay. And remember, when you are preparing the dough fresh, and if you immediately start making bread or baking, you may not see soft and fluffy, bread, right? It's not that soft. Why? What is the reason? Loudly.
The carbon dioxide gets trapped.
Yeah. No, initially I'm saying if you make it fresh and you immediately start baking, would the bread would be a little bit bulky or will not be bulky?
It
It
didn't get
won't.
enough time to rise the dough. It didn't get enough time.
Can you
need to,
could be louder, better, please?
you need to give it some time, or else it will not rise the dough.
Yes, true. You have to give it some time. Why? Because the yeast is now working. The fungus is working to produce carbon dioxide, and that carbon dioxide can be trapped. And of course, ethanol is also produced there. But when you are baking, of course, ethanol is broken down. So it's either removed, right? Destroyed, or evaporated, or damaged, right? So you have carbon dioxide, which is trapped inside. So that is making it little, the dough, the bread is little bulky and soft as well. And you might have seen that you still put some old flour in the fridge, right? And you can even mix a little bit of that. In the baking industry, or even here, I think if you have seen it, people are at homes, so they do keep little dough, in the fridge to preserve it, so that if you make a fresh one, you put little of that one. So that is basically enhancing the efficacy of yeast action, because the carbon dioxide will be produced and the bread will be more softer, right? The bread will be softer. So aerobic respiration also makes alcohol, but that is broken down when you are baking, of course, by the heat. And the yeast is also killed, right? So there's why you must save some of the old thing. So this is again a recap. This question, I'm sure, let's three reason why the microorganism rather than animals are often used in biotechnology. What is the reason? Could you please generally quickly recap? Yes, Hala?
They are small. They don't take up large space. They
Yes,
reproduce quickly. There are
very
no
good.
ethical opposition from people, and they have plasmids which are easy to move, and their DNA have the same basis like us.
Yeah, exactly. The DNA is basic like this. Exactly. All these parameters. So you write it down wherever you want. You need to register all these important points. So who is going to answer the next one? Which product of anaerobic respiration is used to make biofuels? Yes. Yes, Hala, go ahead.
Ethanol.
Yeah, go ahead. Okay. Sorry, you can't hear you.
Ethanol.
Ethanol. Which products of anaerobic respiration is that is used as a biofuels as ethanol? Correct. Yeah, carbon dioxide is taken up by the plants, and ethanol. Which product of anaerobic respiration is important in bread making and in baking industry? Anyone else? Somebody would like to wait a minute. Who is going to answer? Yes, Marush, please.
Carbon dioxide.
True. Yeah, carbon dioxide. And what is the purpose of I mean, what is the advantage of carbon dioxide being produced in the fermentation and baking industry?
It makes
It makes the bread to rise.
It makes
It makes the bread to rise.
to rise, yeah, to expand or rise because the carbon dioxide is trapped. Yeah, okay. So I think this is a practical application of biotechnology. We are using the for the production of various enzymes, right? The enzyme that are needed for different purpose, and as you know, for the washing industries and also in the dairy products, we are producing cheese and yogurt, and also the baking we already mentioned. So they are saying that microorganisms are usually grown in a vessel. We call it fermenters. Okay, so what exactly is fermenter? Fermenter is this is a special vessel. Of course, it has all the requirement to keep the proper environment for the growth of bacteria or fungus. That is very important. You can put nutrients in it. You can supply. You can maintain suitable pH, for example, and proper temperature also, because these are actually the things that are needed for the microorganism to grow. So the microorganism then make the enzyme, and they are releasing in the fermentation liquid, right? The liquid which is in the vessel. And then of course you can purify this enzyme. So when the bacteria are growing, because they are using these nutrients, so you are giving all perfect environment, temperature, pH, nutrient, and oxygen is there. And then they are producing these enzymes that can be isolated. We have these industries that are using different techniques to isolate these enzymes. Now let's see what are those enzymes that we are using these days, the practical application of this technology. I don't know if you can, does it show properly? Oh my goodness, it's not showing properly. Can you look at here? Yes, this one should be okay. Yes, fine, thank you. Yeah, so biological washing powder is, you know, what exactly is a washing powder containing? What is the main constituents of a washing powder?
biological.
I mean, washing powder we are using for the purpose of washing, right? Your clothes, for example, floors and all other general use. So that contains detergent, right? The main thing is the detergent. Detergent itself removing the grease, which is the soap. Soap, what does it do? Soap is basically mixing oil and water so that it can be easily removed from the cloths, right? So it can be taken, it can be washed away with water. So this is one of the reason of the detergent. But we have a biological washing, so that also contains some enzymes. So this is the difference from general detergent, the regular detergent, and biological detergent. So biological detergents are using enzymes such as proteases. What is the function of protease? Breaking down the
Break down protein.
Yeah, exactly. So it's helping. So this biological washing powder is really efficient. Why? Because it can remove the stains. For example, the stain of blood. Blood is containing which protein? Hemoglobin is a protein. So if proteases are in the detergent, what will happen? It can break down the hemoglobin. So this can be, hemoglobin once broken down is soluble in water. So it can be washed away very easily. So this is why we use protease as a biological washing powder to break down the hemoglobin. And then you have lipases, right? Another enzyme that are used as enzyme lipase. And these lipases, as I mentioned, in proteases can be produced by genetic engineering. And they can be easily, because we need large amount of proteases. As you know that there are 10 and 20 kilogram of washing powder packs are available, and there are many, so they are mixing different enzymes. You need large scale production. So to have a large scale production, you make use of biotechnology, and you are using basically microorganism to produce these enzymes. So lipase, as the name indicate, is breaking down the lipids, right? So breaking down the fatty acids and glycerol, and this is good for removing greasy stains. So as you mentioned that, I think this is one of the precautions. As you know that the lipase and proteases, if you are using with an, it can get in contact with the skin, it might damage your skin, right? On regular basis, not on one-time use, it may not be that dangerous, but on regular basis, if you're handling these the detergent powder, it could be toxic to the skin. So they are basically using microcapsules. They are called microscopic capsules. You can see the small pellets. So these pellets are basically covered with a protecting material, and it's not harmful to the skin. So what happens if you mix the detergent with water? So the capsules are solubilized. They are releasing the enzyme now in water. So it can work on stains and different stains that we are using, I mean, for washing cloths or any general use that we are, dishes, for example, exactly, yeah. They have further mentioned that before these lipases and proteases, of course, can work on certain enzymes, sorry, at certain temperature, right? So with a very high temperature, if you're using super hot water, it might be damaged, but they have now made it stable due to somehow they still can work in a very hot or even in cold water. So they have modified by this genetic engineering, as you can see here, even till 40 degrees Celsius, because sometimes you may use hot water for usually recommended because the solution is good, so it can penetrate well, and the cleaning efficiency is increased if you're using a little bit high temperature of water. Is it clear?
Yes.
Okay. I think now pectinase. I'm sure you know pectin. What is pectin? You remember we discussed this probably somewhere, I think, in the germination of seeds and fruits. Pectin is basically a substance which helps stick the plant cell together. So apple, for example, all the fruits that you see, the basic structure is maintained due to pectin. Pectin is insoluble products, and pectins are sometimes used also for constipation. This is why if you're using a raw fruits or raw vegetables, this is a good treatment for constipation and high cholesterol, and it's good to control diabetes as well, because they're not digestible. It gives bulk to the stool, and it satisfies the hunger very easily, so you won't eat much. And it's also preventing the absorption of fats and other, and it also prevents the colon cancer, because many carcinogenic chemicals that are we ingesting by mistake, maybe because there are lots of things that chemicals that we are using in farms. So it will also prevent the contact time with the intestine, with the intestinal wall. So colon cancer is also reduced if you're using high fiber intake, high fiber fruits, for example, vegetables or salads. So pectin is making the structure of the fruit, okay? A fruit such as an apple or orange contain lots of pectin. If the pectin is broken down, it can be much easier to squeeze juices from a fruit. So if you break down this the pectin, the fruit will be very easy to squeeze. So you can get maximum of juice. And this is one of the techniques that we're using. You are having the artificially available, the juices that we are using. They are using, of course, natural fruits, and they are saying we are not using 100% natural, but still they're using chemicals to break down the pectins, right? So pectin is also giving, it is like dissolving the whole material. If you put apple in the pectinase is an enzyme that will break down the pectin, right? So the whole apple will be dissolved, it is just mixed, and it will release lots of juices, and then you filter it out. So this is how we are using this commercially for to produce fruit juices. That's why squeezing the fruit is much easier if you are using pectinase. So as the name indicate, ase is breaking down of the pectin. So I think this is for the extraction of fruit juices, nothing here. So let me see what they have mentioned for other. Okay. So now we started with, we are now discussing the practical application. As you mentioned, the washing powder, and then you're talking about the pectinase itself. And now we're coming to lactase. I'm sure you know lactose is the milk protein, remember? So it's available in milk. So lactase is an enzyme that is breaking down lactose, yeah. So the sugar which is found in, this is the sugar which is found in milk, right? So lactose is converted to glucose plus galactose. So remember that there are certain, not only babies, but even adults, they have this lactase deficiency. In the initial stage, of course, for 2 years, they are mainly dependent. Babies, newborns are dependent on milk, right? So how they can digest this milk? By releasing lactase. in the intestine, right, in the digestive tract. So in that, if digestive system is not releasing lactase, lactose cannot be digested. So they will have lactose intolerance. We call it lactose intolerance. They cannot tolerate. They rather will cause diarrhea and flatulence and also, you know, bloating, which is really problematic. So some people, for example, those in the European, they have mentioned is more, and if we have seen even in Asian, they do have lactase deficiency. They cannot use milk or dairy products very easily. So what basically, not like normally, yes, those people we are using yogurt, we are using laban, milk, we do have lactase, it can be released, and that is how we can break it down, right? But those people who have deficiency of this enzyme, of course, they cannot tolerate milk. They cannot take it. So milk can, but for them, milk can be treated with lactase to break down lactose. So that people who do not make the release of lactase, they can drink milk or they can eat these products very easily. And we do have lactose-free milk available. Remember, lactose-free milk is comparatively expensive. Or you could, if this is a minimal problem, moderate problem, not severe, so probably you don't need to use the lactase-free milk, lactose-free milk. Rather, you can use lactase enzyme on and off. So this can also help. But of course, if you need lactase enzyme, so you need biogenetic engineering, right? Biotechnology to produce more enzyme. Otherwise, you won't have that much amount to use it for to work on the milk and then use it commercially, right? Okay. So what about penicillin? Everybody, I think you are aware of the penicillin, right? Let me see what we have discussed here. We have discussed the making bread and making enzymes, and then pectine is finally coming to penicillin drug industry. Okay, as you know, penicillin is antibiotic, and this antibiotic is very common. What does it mean? What is antibiotic?
Antibiotics.
Medicines which are used to treat bacterial infections.
Yeah, they are used to treat bacterial infection like pneumonia, skin infection, acne, for example, tonsillitis, and you have different conditions. Yeah, they are antibiotics or those substances, those chemicals that can kill bacteria. Okay, and those bacteria, they are really pathogenic, or they can cause disease. So penicillin is, you know, this is produced by a fungus. And that was also an accidental discovery. I think you can go to the discovery. This story is very, very interesting. It was done in a laboratory by a scientist who accidentally saw that one of the culture media where he was growing some bacteria. He was a microbiologist working with some strains of bacteria he was growing. Then one day he found that there was zone of inhibition. The colonies were not growing well. And then he found the yeast, actually, there was contamination of yeast. Fungus was growing in that area. And that area was, bacteria was not able to replicate in that near the area surrounded by yeast or fungus. So that is called penicillium. This is called penicillium. The name of the fungus was penicillium. And that's how they have named it penicillin, right? The drug name is penicillin because it's produced by the fungus penicillium. So I think they have given the production process also probably. They have discussed it. Let me see if they have given anything. Yeah, here's the, yeah, okay. So this diagram, they are saying this is the fermentation unit. We also call it fermentation. You could see here the vessel itself is containing what? So this is containing the culture of the fungus, okay? Penicillium, containing carbohydrates and amino acids. The carbohydrates, as I mentioned, is again, this fungus will work on. This is a nutrient for them, right? And you are also introducing sugar for this one, right? Penicillium and sugar constantly, because you know a part of fungus is already here, and you're constantly rotating so that the fungus can mix and it can get fresh nutrients for the growth. And you could see it's jacketed, so you are controlling the temperature also. So you need a good temperature, you need nutrients here, you also need bubbling of oxygen, which is required here. So there is also mixing and also required for the for the fungus to grow and ferment, right? to do the fermentation. So what they have discussed, the whole thing about this assembly, that how you will control, initially you need fungus to grow. Of course, you need, you let some time, which is, they are saying maybe 15 to 24 hours. That is the usual time for the fungus to grow here. When the fungus is growing, it's releasing penicillin in the environment. So here, penicillin is also produced at the same time when the fungus is growing. So, but they are saying you should not put lots of sugar here. This is gradually added. The sugar is added in a gradual way. If you are putting so much sugar here, the fungus is not going to grow well. If you are giving very less sugar, it still would not be producing penicillin very well. So that is why you need to control the sugar. They have done experiments for years and years, and finally they have decided what amount at what time we need to introduce. Of course, this fungus is constantly growing, right? So they are making use of these nutrients that are available. They are consuming. So of course, the whole thing is consumed finally, right? And you need to replace it. That's why there are certain days. Maybe you can continue this fermentation, this process, maybe let's suppose for 1 week, and then you need to remove the whole thing. You need to add everything fresh, because it does not go for longer. So once, I mean, for 1 week, for example, you could still isolate penicillin. So you can collect the environment, the liquid, you could collect it, and then there are way of purification of penicillin. You are producing pure penicillin. So then this penicillin is a basic structure which is sent to different industries. So they are making range of penicillin antibiotics. So we have amoxicillin, as I mentioned, ampicillin, cloxacillin, and augmentin, there's amoxicillin, right? And ticarcillin. There are different names, but the structure is the same parent compound. The original penicillin, which is produced by fungus, is then stored and sent to different industries. The industries are making different kind of penicillin. For example, in Pakistan, maybe they are using amoxicillin more, but we don't need, maybe cloxacillin is more common in America. In Saudi Arabia, we may be using ampicillin. So that's why they, tailor the needs. They look for what exactly is more needed in the market, so they can make that penicillin. And I think they have just mentioned these things, that you need to take good control of the temperature, you need to give them enough time, and then you need to, the sugar contents, the availability of sugar, and control the pH as well, okay? So now we are coming to, any question? Is it clear, this for penicillin production?
Yes, sir.
Yeah, okay, to recap just that, this is the fungus which is grown in a particular vessel we call fermentation tank, and it has got nutrient, oxygen, is providing all the requirement for the fungus to grow. When the fungus is growing, it's naturally producing penicillin. So that's why there's a specific time, maybe 1 week or 10 days, then you isolate the penicillin, the chemical itself. You can isolate it from here. So now coming here to genetic engineering, as I mentioned, biotechnology, genetic engineering is common these days for biological products. We are using different vaccines, we are using different enzymes, we are different biologicals that are needed to treat diseases. One of the common thing that we are using is insulin, which is needed on regular basis for type 1 diabetes. You have type 2 diabetes. In type 2 diabetes, you still have some insulin, but there's insulin resistance. The insulin is not working properly. That is why we are using tablets for those patients. Type 2 diabetes, we are using tablets that will help to overcome the insulin resistance. Insulin resistance could be due to many reasons. One of the major factors is obesity, right? Obesity is causing insulin resistance in diabetes. But type 1 diabetes is not producing, pancreas is not producing insulin at all. No insulin. So that's why it's must be injected. Insulin must be injected for the, this is important for the patient's survival. They won't be able to make use of glucose. And that would definitely, patient cannot survive. It will cause many complications to the renal system, to the eye, brain, and cardiovascular system. So we need insulin. And insulin, of course, if we are isolating from the pancreas, it's again a very laborious process. You cannot get insulin in large amount to be administered to patients. So what is the proper way? They are basically using insulin gene. Human insulin gene is inserted into bacteria. And I mentioned, I have sent to you today, I think, one of the figure of the basic technology of genetic engineering. How do we do that? And they have also discussed it, I think, here. So we'll go through it. So that gene is introduced into the bacteria, which is, I think, E. coli, the name of that bacteria. And this bacteria is replicating very easily, producing insulin. So this insulin is now isolated and commercially available. We have different forms of insulin. And we are also using crops. The genetic engineering can also be used in crops, plants that are genetically modified. You can modify the plant to be resistant to herbicides or insects or some disease, right? Because if you're, for example, cotton crop is very easily damaged by insects and by all the pests. So how to control that? Either to use the spray, right? If you're using the spray, it might be toxic to the crop itself. So what basically you do, you are making use of genetic engineering. You bring some changes in the seed genetic material, so that it grows, the crop will be resistant to that particular disease, for example, or they will be resistant to certain pesticides or insecticides that we are using. So there's why genetic engineering is changing the life. There are also pros and cons, of course, advantages, disadvantages that we will discuss here. So can you, yeah. So they are talking about this genetic engineering using in the plant industry and in agriculture. So that you make the crop in a way so they are not harmed by the herbicide, which is called glyphosate. Glyphosate is the insecticide which is sprayed on the crop. So it might be damaging the crop. So by using genetic engineering techniques, you can modify the plant. You can modify the crop to be resistant to this insecticide. This means that the farmers can spray this insecticide without any problem. They won't be causing damage to the crop itself. Rather, the weeds will be killed. I think in the cotton plant industry, they have genetically modified so that they contain a substance which is called Bt. This is also a bacterial toxin, okay? Bacterial toxin. This is Bacillus, I think Thuringiensis, something, this is a name, but this is a bacteria through which they have incorporated that gene. So what will happen? So if you have genetically modified crop for the cotton, so the cotton plant is producing a toxin. So when that toxic material, when the insects are feeding on the crop, so they are destroyed, because this is toxic specifically to the insects. So that's why in this way you can get rid of the pest. You even don't need to spray pesticide or insecticide chemicals. This can also be by genetic engineering. So they have taken this Bacillus microorganism, they have taken that gene which is producing a toxin, and they have introduced that genetic material into the crop. And that's why crop in its growing, it is producing that toxin. And when insects are feeding, so they can be easily controlled. Like, for example, they have mentioned this ball weevil are killed if they are eating the cotton plant. And this will also reduces the use of pesticides. I think I'm sure it's clear to you. And they are using this another. Keep this example if you're asked about the application of genetic engineering. So this is one of the example that, for example, in drug industry, for example, producing insulin for type 1 diabetes, and then producing some resistant crops. They are resistant to either pesticides, or they are resistant to, or they are producing some toxins that will kill the pests or the insects that are feeding on that crop. And they are saying that there's a golden rice production is also to have more vitamin A. Remember, so normally the regular rice contains vitamin A, and vitamin A has got very important function in your body. What is that? What is the role of vitamin A? For your eyesight, right? So deficiency of vitamin A will cause blindness. So they are saying that golden rice has got more amount of vitamin A. So those people who have vitamin A deficiency, we also have carrot, it is added in the milk also, and dairy products. We do have, these supplementation available in the market. But those people who have severe deficiency of vitamin A, they can make use of this rice, which is called golden rice. And this golden rice is not a normal rice. This is genetically modified. So you can control the traits, you can control the properties or quality of certain things in the crop by using genetic engineering. So they have modified the crop in a way so it can produce more vitamin A, which is good for people who are having genetic deficiency of vitamin A. Okay, now they have actually a long debate here that genetically modified, this is called genetically modified plants, is it good or bad? For example, in case of insecticides, people are saying that if you're producing BT cotton, for example, it's possible that human will have exposure to this toxin. So it's possible it could be damaging to human as well, right? Or they are saying if you're using, for example, the weed, sorry, what was the name of this insecticide? Glyphosate. Could yeah, this is, yeah, glyphosate. If you are using a crop that is resistant to glyphosate, right? So it's possible that this resistance might be transferred to weeds. Is really? Do you think it can be transferred to weeds? They have a discussion here. You can read it in detail. Yeah, exactly. Weeds are different, but there is one thought process that it's possible that this might be transferred to weeds and you will have super resistant weeds. So there's why instead of this pesticide or herbicide killing the weeds, it might damage the crop. So this is one of the disadvantage, and especially the BT, maybe some of the toxin that are produced have exposure. A human can also have exposure, and that could be damaging. So far, of course, we have not seen any such problems, but yes, they are not the natural way. As you can see, the size of the fruits these days, and especially the genetic engineering tools that we are using in dairy farms, you know, the farms, chicken that are producing, they are grown up in just few days or few weeks. And the milk production that we are using genetic engineering techniques, drugs and other stuff, and we are producing breed to give more production rather than quality. So of course it's going to affect the quality. As I mentioned about the banana before, the size of banana and the wild banana and also the genetically modified bananas, definitely there's a big difference. There are a lot of debates on it. Some are really good for certain categories, certain population, but at the same time, each aspect, I mean, this process has advantages and disadvantages. So that's why organic food, you know, industry is also common, is very expensive, is growing again because people have cancer and other diseases, and they are old aging, you know, people are getting accelerating the age, and people have psychiatric problems, cancer, for example, and other stuff. And they are also saying that, for example, beta toxins, I think they have mentioned another thing. Yeah, there's some of these genetic engineering methods would also damage the ladybugs, for example, ladybirds and other predators that are feeding on insects. If they will eat, they will also die. So this is another way, which is might be, these toxic might be harmful for them. Okay, because if they are eating on the BT toxins, they will harm, of course, the those predators. So do you have anything in your mind you would like to share about this process, genetic engineering? You have heard, you have read about it? Yeah, okay. Today, I think, so these are the different processes that I discussed with you. So make sure that you remember what exactly is genetic engineering and what is the use and what are the advantages and disadvantages. And how do we do the genetic engineering here, okay? So this is the normal way of genetic engineering. You could say a baseline technique. As you know that, if you remember, I also mentioned in the, I sent the picture in the group. So you could see the plasmid, which is the, other than the chromosomal DNA, this is a ring form of the DNA, which is present in the bacteria. So that can be used to transfer the gene. So what basically you do, for example, this is the human cells and you have special gene. You want that to be produced by the microorganism. So you take that human cell, you are liquidifying, you are digesting the cell to extract the DNA, because the cell needs to be split, digested. This is called splitting or digesting of the cells. And then you are precipitating the DNA, right? This is the second step. You add some chemicals because now this solution is containing, of course, DNA, and then you have precipitated the DNA. Now, third step is the restriction enzymes are added to cut the DNA into pieces. What are these restriction enzymes? What do you think? What does it do? They are saying that they are cutting down the pieces of DNA. So they generally cut all the DNA or specific part. Anybody would like to share your thought with me, please?
Specific restriction enzymes cut specific genes and the codings.
Very good, exactly. This is the main use of restriction enzyme, not to cut to split the whole DNA, but that's why we have different restriction enzyme, which is targeted. If you want the gene for insulin, you have a special restriction enzyme that will split that part of DNA which we need to incorporate into bacteria. Okay, so it's splitting and producing sticky ends. Why sticky ends? Because of the nucleotides, right? Like you have adenine, guanine, cytosine. So you have the same one that will join adenine with the thymine or guanine with the cytosine, for example. So these are sticky open ends. They are produced, right? And restriction enzymes are also used for the plasmid. This is the plasmid, okay? The plasmid is the circular ring, which is a DNA part, but it's not the chromosomal one. It is present in the bacteria. It's work like a vector. We can make it recombinant. So restriction DNA has cut the part which we need from the human cell, right? From the human DNA. We want that particular gene to incorporate into this bacteria. So we have got this piece, and then we have split this part. So now this is open end. So what will happen? You will have enzyme ligase, ligation. This guy ligation, this guy ligation. Ligation means binding together. So if you bring this part in these plasmid together, so it will cause the recombinant plasmid. You recombine. This is not the original one, right? A part of it from bacteria and part of it from the human cell. This gene you want to be incorporated here. So now this is a recombinant plasmid, and you put this recombinant plasmid in with bacteria. You want that bacteria to grow. So this bacteria will take up this plasmid inside. So now this bacteria is replicating. When it is replicating, actually it is producing whatever gene you have incorporated. Let's suppose this was insulin. So you put and replicate the whole environment. You can see the fermenter here. This is the vessels where you can do the whole culture. You are giving chance for the bacteria to grow here. When bacteria is growing, you're providing all the nutrients, okay? So it's producing now insulin or whatever enzyme, whatever is your target. So it will be then isolated. So this is called genetic engineering, and this is called vector also. This is carrying your selective gene, right? So you keep in mind the role of two things. One is your initial, the gene that you want, the human cell, for example, pancreas cells you want, right? So, and then you have the restriction enzyme, and then you have plasmids, okay? So, and here you need to ligate, therefore ligase. Restriction enzyme and ligase are working. Restriction will cut the pieces of DNA of both plasmid and the human DNA to cut that part, and then you do the ligation by using ligase enzyme, and then you put this plasmid with the culture of bacteria. So this bacteria will take up the plasmid, and then you allow this bacteria to grow on a large scale to produce that particular gene. Is it clear?
Yes, sir.
Yeah. Any question, anything? Otherwise, we'll go to end chapter question, inshallah. Okay.
No, sir, no questions.
Okay. All of you. Is there anyone else? Anything you want to add up? Please go ahead. This is your session, inshallah. Don't worry. Whatever you want to comment or what question comes to you, just please go ahead. All right. So let's go to end of chapter questions. So what are the question number 1? So who is going to read it and who is going to answer?
Can I read?
You want to read? Sure, go ahead. And if you can explain a little bit, that would be great. You can go back to the book pages. You can read it. It's okay, inshallah.
Copy and complete these sentences using the words below. You may use each word once, more than once, or not at all. Yeast is a single-celled dash.
Okay. dash. Okay, so you can pick up from the yeast is a single celled dash. So it is amylase, it is bacterium, it's a biofuel, it's a bread making, it's a catalase. Yes, Hala?
Fungus.
It's a fungus. Very good. Yeast is a single celled fungus. As I mentioned, we're using microorganism, yeast and bacteria. Okay. So single cell fungus, it can respire anaerobically. What is anaerobic respiration? What does it mean?
In absence of oxygen.
Absence of oxygen. Very good. Okay. So breaking down glucose to form
Ethanol.
ethanol. Okay. And the gas?
Carbon
dioxide.
Carbon dioxide. Very good. So yeast, so, and what do you call this process? If they ask you, the process is called
Fermentation.
fermentation. Very good. And what is, where we exactly use this process?
To make bread.
Yeah, baking industry, bread. Very good. Excellent. Okay, next.
To make ethanol for use in dash. Using biofuel.
Using as a biofuel. Very good, because ethanol is a biofuel provided with the sugar that have come from crops such as maize. Yeah, we mentioned maize. Remember, is the alternative to which fuel? Which fuel?
Yes.
Fossil fuel, gasoline. Yes, exactly. Yeah, we are using basically yeast for that to cause the fermentation. So that fermentation is producing ethanol that can burn very easily. But as I mentioned, ethanol is not a good source of energy. The energy will not be enough for the engine to carry or drive the car. So, okay, next.
Yeast is provided with sugar that have come from crops such as maize. To make bread, yeast is mixed with flour and water. The enzyme dash break down starch in the flour.
Okay.
Amylase.
Amylase, very good. Yeah, we mentioned about that. Exactly. Amylase. Okay. Breaking down the flour,
producing a dash that the yeast uses in respiration.
Yes.
Glucose.
No, not sucrose. We mentioned glucose and maltose, right? Glucose, producing glucose that the yeast uses in respiration. Then you remember the yeast is working on glucose then. So this forms
Carbon dioxide.
exactly. This forms the carbon dioxide, which is making the bread rise or expanding, right? Because the carbon dioxide is trapped. Shabbos, very good. That's excellent, mashallah. So you understand this point. So this is the practical application also. Whatever we have discussed in the chapter, so they are revising all those concepts here in the form of question. So we can move on to the next question. So what is the next question? Yeah, okay. Yeah, okay. Outline the role of each of the following in genetic engineering. Okay, so you need to understand the role. When we say the role, we mean application. Okay, application of these. So what is the role of or application of restriction enzymes? Yes, Hala?
They cut the required genes or the bases in both the human DNA and the.
Okay, can you please repeat it a little louder?
They cut the required bases or the genes in both the bacteria plasmid and human DNA.
Yes, very good. To cut the particular part of the DNA or gene that we need, and also to cut the plasmid where we want to incorporate that particular part of gene. Okay, yeah, this is the function of the restriction enzyme. So both, and how they can do that, you can also add something else. They just cut it. I mean, if you cut it, is it double helix or the part which is cut, the ends? That's why they call it sticky ends. Yeah, it's on the saying they are producing sticky ends. So it means, what does it mean? They are not this single strand. Okay, the nucleotide are exposed so that it could be easily joined, the one which is there in the plasmid. If they are not sticky ends, of course, it's not going to work, right? Ligase can only work if they are. So also add that they cut the target gene from the DNA, and then they cut the plasmid and they produce sticky ends of that particular part of the DNA. So what is DNA ligase? The role of DNA ligase? Who is going to answer that? Ligase, is the name indicates, causing ligation. Ligation at tying. You tie something. This is called ligation. So ligase. What will be the function of ligase?
It links the sticky ends together.
Very good. It's bringing the sticky ends together to repair or join the part of the target gene and to the plasmid, right? Yeah, this is the enzyme ligase. This is the enzyme which is required for the ligation or tying up something. You know, this is okay. What about the plasmid? What is the role of plasmid? What does it do? As we mentioned, the circular DNA material in the bacteria. So what does it do? The restriction enzyme is cutting also plasmid in the target gene, right, from the DNA. So the plasmid is basically used to transfer that particular gene into bacterium. This is called the vector, right? And this is called recombinant plasmid. The role of plasmid is to transfer that gene. If you cut it from DNA, how would you transfer? You have to ligate it. You have to fit it in the plasmid, because plasmid can be easily taken up by the bacteria. You cannot inject that piece of gene directly into the bacterium. If you inject it, it's not going to work. So it has to be compatible with the bacterial system. So to be compatible, we want to put that part in the bacterial plasmid. So you got my point. Anyone would like to repeat it again, please? Could you repeat? Plasmids, what will be the role of plasmid? Yes. Can you explain the role of plasmid? You didn't understand the plasmid?
They are the main DNA part in the bacterium.
Okay. This is the circular part, yeah, right? So what will be the role of, now you can see the role. What is the role of this plasmid? What does it do? This is the main
They
player.
are cut and they join with the DNA, human
Yeah,
DNA.
so yeah, this is the procedure. The procedure is to cutting plasmid and cutting the part of DNA. But the role of plasmid is to transfer this particular end, you know, this particular gene to bacteria. So this is helping and transfer. The role is to transfer your gene of interest, the one that you want bacteria to produce, that protein or this enzyme you want to produce. So that is transferred through the plasmid. So plasmid is working like a carrier for that particular gene to be incorporated into bacteria. And now this bacteria is now then replicated. Okay, so this is the role of plasmid. Okay, so let me, is it clear, inshallah?
Yes, sir.
Okay, very good. Let's see. Number 3, selective breeding and genetic engineering can both be used to improve the yields of crops plants such as maize or soya. So A, explain the difference between selective breeding and genetic engineering. What comes to your mind? What is selective breeding and genetic engineering? This is really, I think, question which may be related to common sense rather than scientific explanation. Go ahead, please. Selective breeding and genetic engineering. Yes, Hala?
This is a process in selective breeding is a process in which the advantageous species of animals or plants are bred among themselves, and the genetic engineering involves the incorporation of the human DNA with the bacterium.
Yeah, genetic engineering is not related to human. Yeah, it could be also possible in plants and other species, right? So, but yes, you are right that in case of selective breeding, we don't transfer the genetic material. We rather took parents, right? The parents that we want, for example, tall or small, for example, in case of goats or sheep or cows. So we breed them together. We don't do any genetic manipulation. While in genetic engineering, we take particular gene and incorporate that in another species, right? So maybe the same, yeah, the same species, but we incorporate a particular gene of interest. right? This is called genetic manipulation. So you are, but here you don't change the gene at all. You just change the parents, you know. So like, for example, if we, there's a white goat and black goat, for example, male and white and black female. So the offspring will also be different characteristic. And you want, for example, some goats that are producing more milk, and then you breed it with another goat, different speed, right? You can say trait or different qualities. So you can breed them together. So the offspring that they are producing will also carry some of this genetic, you know, trait from each other. So there's a mix and match of the genetic material. But in genetic engineering, you are basically taking exactly that genetic code, and then you are introducing. Okay, excellent. Yeah, this is the explanation. Okay, so what is coming? Outline one example of use of genetic. Yeah, genetic engineering, remember, is a newer field. It's the novel way of manipulating things. Selective breeding is old, right? So this was introduced maybe in 70s, where we were able to know the genetic mechanism, molecular genetics. The scientific discoveries helped us to understand the genetic material, genetic process. And this is how we are using this technology now. Yes. Okay, outline one of the example for the use of genetic engineering to increase yield in a crop plant. Okay, they are not talking about those human products now, they are talking about the plants, agriculture. So would you like to give some example? We mentioned two examples here in the discussion before, remember? One was the cotton plant, another was the golden rice. So you can, if you are producing BT crop, BT cotton, with the bacillus toxin, so you are increasing the production of the crop because they will not be easily damaged by the insects, right? So they will not be damaged by the pests. So this way you can increase the production. And what about the golden rice? What was the difference in the golden rice and normal rice?
There's more vitamin A in the golden rice.
Yes, yes. So this is also genetic engineering, right? You are introducing some genetic properties in that crop to produce more vitamin A for people who have severe vitamin A deficiency. So golden rice is another example, which is producing more crop with vitamin A production in crop industry. And also you could also use maize and many other examples. Okay. Discuss the possible advantages and disadvantages of example you have described and B. Yeah. So you mentioned this crop and both the bee toxin, BT crop of cotton, and then you are also talking about the golden rice. And also the herbs, you know, the pesticide, the genetic engineered crop which is resistant to the pesticide. So this pesticide cannot damage the crop, it will damage the wheat selectively. So what will be the advantages or disadvantages? The advantages is, sure, you already mentioned, the crop production will be increased. Weeds will not grow easily, right? So you have more production. What will be the disadvantage?
The BT can cause diseases in human being if it gets ingested.
Yeah, if it can be adjusted. And also remember about the predators, if they eat on that, so it might kill those predators. That could be toxic for them as well if there is any exposure to human. And they are saying it may be possible that BD toxin might be reduce some of some of the resistant human pathogens, the resistant pests or insects. So it's, by gradually they might spread this problem to other crops also. So it may be one of the disadvantages of that one. And golden rice is also another example you mentioned. And of course, if you're using too much vitamin A, so probably it could be toxic to human as well. So if for example, it is used for non other than medical purpose, those people who do not have any deficiency of vitamin A, if they are using that rice, they might have toxicity of vitamin A. So this is one of the things that you should keep in mind of these advantages, disadvantages. So whatever example you are giving, you need to mention the advantages and disadvantages. Advantages are very clear, they will increase the production and fast replication and safe and stuff like that. But for the disadvantages, exposure to human, exposure to the predators, for example, and it might be spread to other crop. Okay, what about the enzymes are used commercially to extract juice from apple, right? So the diagram below shows there are two containers of apple juice, one containing juice extracted using an enzyme and the other without an enzyme. Okay, this one is using enzyme and this is not using enzyme. So A, with reference to the diagram, compare the volume and appearance of the two juices extracted. Yeah. So first of all, look at the volume, not using the enzyme and using the enzyme, and then explain it, please, also. Hala, go ahead.
The juice which is extracted using the enzyme has the volume of 15 centimeter cube, and it is less of a cloudy, and the one which is extracted without using the enzyme it is less in quantity, 6 centimeter cube, and it is cloudy also.
Cloudy also. So the reason is why? Which enzyme and why?
The pectinase has not broken down the pectin.
Very good. Yes, the pectin was not there. So pectinase, as I mentioned, can also increase the volume of juice. It can easily, you can easily squeeze the food. So that is why it's easy to make more juice, to increase the production of juice or extraction of juice. And it's also making this clear because you can easily filter it, but this is really still turbid. Yeah, because you are not using the enzyme, the pectins are not broken down. Okay. Now, can you read please this one? Somebody can read it and then try to understand. Okay. This is an experiment. Okay, go ahead. Yes, Hala?
Certainly.
Okay.
Some students investigated the effect of pH on the production of apple juice using the same enzyme. The apples were chopped up and formed into a smooth pulp. The pulp was divided into samples. Each sample was adjusted to a different pH. Enzyme was stirred into each sample of pulp and left to stand for 10 minutes for the enzymes to react. The mixtures of pulp and enzyme were then filtered for 20 minutes to collect the juice. The volumes of apple juice collected are shown in the table.
Yes, exactly. So would you like to explain this experiment? So this was an experiment conducted. So first of all, they wanted to see the effect of, what is the objective of this experiment? To see the effect of
pH on the apple juice.
So the pH, the effect of pH would be related to the effect of enzyme, right? So how enzyme react in different pH. If you change the pH, what will happen to the activity of enzyme? And of course, if enzyme is not working, you will also decrease, you see the decreased production of juice. So this is the objective, is to see the effect of different pH on the enzyme activity in producing apple juice. So here is the table. Okay. Okay, let's see. Here's the table. Okay, now you could see here the pH now is 3 and the volume is 40, then 40, then 56, and this, I think it's maybe by mistake 3, 4. This is 5 because you can see it's in sequence 3, 4, 5. So it's not 4 because there could not be the same value. 3, 4, 5, 6, 7. Okay. So now construct a graph to show the effect of pH on the production of apple juice using this enzyme. First of all, what does it show? Immediately looking at this data, what does it tell you? Don't answer this question one. Just look at the table, the data. What information you can get from this table? Yes, go ahead, Ada.
That as the pH is increasing, the volume of apple juice collected is also increasing. But as the pH reaches 7, the volume starts to decrease.
So what is that pH? What do we call that value of pH where you get the maximum production of juice? What do we call it?
Acidic.
Optimal pH, right? Where the enzyme activity is maximum. So you are in pH is 3, little effect. You further increase the pH, you increase the activity. And then up till 5, the pH is increased, right? The juice is because the enzyme activity is increased. That's why you have more volume of apple juice. So till 5. Or what happened to the enzyme when you are further increasing pH 6 and 7? Why it's decreasing?
Because it's
The
becoming
enzyme,
Because, yeah, go ahead, please. What is that?
the enzyme
Enzyme is
activity is slowing down and it might become denatured.
might become denatured. It might be denatured. Very good. So enzyme is, as we mentioned, work on specific pH. If you are changing pH too much, it will damage the enzyme, right? It may not be favorable for enzyme to work in that particular pH. So it needs a certain value of pH. So that is why you have at 5 the maximal activity. If you increase it further, the enzyme is losing the efficacy. So it's not going to work. Yeah, so construct a graph. I think if you, I just will show you here, how would you construct a graph? So you have a data table, right? You have two values. First, look at the values. Not sure it's really quick. Cannot decrease that. Yeah. Yeah, okay. This is your pH. Yeah, that's good. So on one x-axis, you can have pH values. So you can choose either 2 boxes representing 1 or 1 box representing 1. You can choose any value you want. So I mentioned 1, 2, 3, 4, 5, 6, 7, right? So at 1, the effect was 40. The volume was 40, right? Yes, Hala, go ahead.
Sir, how do we know which of the values is independent and which is dependent?
Okay, we'll come to that, the environment. Yeah, so which value you need to constant and what are those factors that will affect it, right? So basically pH is the one that will affect the effect of enzyme, right? So, but here you can just plot the efficacy, construct the graph so that you could easily compare, I mean, which value, at what value the production, this is the one, which is the output. This is the output of enzyme. So you cannot measure the enzyme, you can measure the effect of enzyme. So what is the effect of enzyme? The production of juice. So that is dependent actually activity, that it's depend on the pH. So you stand from 1, 2, 3, 4 till 7 and then plot it 40 and then you have, I think, how much is the 50? 56 or 60, yeah, right? And then you have 95 and then it's coming down to 56 and then finally to 30. Yeah, so here, so you could see here, if the moment you are increasing the pH value is gradually increasing the production of juice, so until it reaches maximum. So beyond this point, if further increase, you won't see any further increase, rather it's decreasing, right? So you could see this is the pH and then it's okay. Just attach it. Yeah. Okay. Check that. So is this graph clear here? So in the next question that you are discussing about those factors, then what factors you need to make it constant so that the reaction is dependent, right? The reaction is, the experiment is properly organized. Otherwise, there will be lots of variation, okay? There are certain variables that you need to keep it constant, because if you make slight changes in those parameters, you may not see, because you want to see here what happens if you change the pH, okay? You are just, this is the variable, this one you want to change. And this one, the rest of the thing you need to keep it constant. What is that? What are the things that you would like to keep it constant, for example? Yes. Yes, Anna?
The amount of apple pulp used or
Very good.
the
Yes,
enzyme
amount.
added.
Amount of apple pulp and amount of?
Enzyme added.
Very good. Amount of enzymes. And what else? Temperature. Remember, because if you're increasing temperature up to a certain level, you might increase enzyme activity. Even if the amount is less, you still can see greater effect up to a certain level. Enzymes are also temperature and pH dependent. So amount of pulp, the raw material that you're putting, if there are slight changes in it, that would also affect the production of juice. And the pH, the amount of enzyme, of course, should be fixed. It should not vary. And temperature, these three variables, you need to keep it constant. The only thing which is changing is the pH, okay? So this pH is basically affecting the enzyme activity. So the effect of enzyme is dependent on the pH, okay? And how would you know that enzyme is working? To look at the production of the juice. So this is the thing that you need to. This is, I think, the last one, then probably we are done. What is the last question? Yeah, you already mentioned the optimal pH, that is 5, right? They have mentioned, I think there's some typographical mistakes here, is 4, or do you make it consider as 5? Describe and explain the effect of pH and the production of the apple juice using this enzyme. Would you like to explain? You already answered this question, okay? You have controlled investigation. What factors you need to control? Temperature, amount of the raw material, the pulp of the apple, and the amount of the enzyme. And then what about this? Describe, explain the effect of pH on the production of apple juice. Would you like to explain that, please, from this data? What comes to your mind? Yes, go ahead.
As the pH is increasing, the volume of apple juice collected is increasing until the pH reaches the optimum pH, which is 5. The production, the volume of apple juice then decreases.
Very good. Yeah, this is the explanation. This is the exact scientific explanation. Excellent. That the effect of enzyme is dependent on pH. As we increase the pH, gradually it's increasing the enzyme efficacy. That's why we see more volume of the apple juice. And this enzyme, you can write it down, maybe pectase, right? Pectase is the enzyme that we are using. So, but after a certain pH, the activity of the enzyme would be decreased. So that's why enzyme has got optimal pH. So it won't work beyond that limit. And it has specific pH where it has maximum effect. That's it. Do you have any question, anything?
No, sir. No.
Is it clear, inshallah?
Yes, sir. Yes.
Okay, which chapter do you want tomorrow to cover for the exam? Which one do you think is important to be covered? Then let me know in the group if you want, which chapter do you want to cover? And do you think the timing will be okay, 9 p.m. or you want it in the daytime? Because tomorrow you, I think on Sunday you have exam, right?
Yes.
So do you want to take it during daytime, afternoon, sometime like that? Is it okay?
Sir, this time is okay. Sir, daytime is better.
Daytime is better. Okay, inshallah. So I'll let you know, inshallah, tomorrow. Okay. Thank you so much. Jazakallah khairan. Assalamu alaikum wa rahmatullah.
Wa alaikum assalam.
Yes, sir. Allah Hafiz.
Allah Hafiz.